Good day, everyone. I'm Mr. Ong from Sriputra Sixth Form College. Today, we would like to talk about light independence reaction, which is known as Kelvin cycle. Previous lessons, we have learned how to explain the non-cyclic photophosphorylation and the cyclic photophosphorylation. Today, we would like to describe light independence reaction. In light dependence reactions, the energy of the ATP and the reduced NADP that produced is used in the formation of organic molecules from carbon dioxide. As you see in this slide, this is a chloroplast. Then enlarge it, this is a granum. So for thylakoid, so in the thylakoid, you can see here, this is what you have learned in the previous lesson, which is a light-dependent reaction. The ATP and reduced NADP that form or that produce will be used for light-independent reactions to produce organic molecules, especially glucose. Light-independent reactions, also known as Kelvin cycle, which is occurs in stroma, that through the 13 reactions. However, compared to Krebs cycles, these Kelvin cycles, the 13 reactions, you will not need to know in detail. The 13 reactions of the Kelvin cycles are divided into 3 phases, as you see in this diagram. First is the carbon fixations, followed by carbon reductions and regeneration of RUBP. RUBP is ribulose bisphosphate. Since there are 13 reactions, therefore there are 13 enzymes that requires to catalyze each steps in the Kelvin cycles. And then all the 13 enzymes are found in stroma. Carbon fixations is the first phase in the Kelvin cycles. Consists of a single reaction, which is only one reaction, in which a molecule of carbon dioxide, this is 3 carbon, okay, one sphere here represents 1 carbon atom. So in which a carbon dioxide react with a phosphor-related 5 carbon compound. So this is what we call the 5 carbon compound, 1, 2, 3, 4, 5. So this is known as ribulose bisphosphate. So to form an unstable 6 carbon compound. But the unstable 6 carbon compound, since it's unstable, so it's immediately split into 2 molecules of phosphoglycerate. So this phosphoglycerate, or we call PGA, is a 3 carbon compound. So this reactions is catalyzed by Rubisco. Rubisco is the short form for enzyme ribulose bisphosphate carboxylase oxygenase. So here, this enzyme is a bit different. So since it is a carboxylase, at the same time, it is also an oxygenase. That means this Rubisco can be used to fix the carbon dioxide or can be used to combine with oxygen. Rubisco is activated by light and are more active at the pH 8 and require magnesium ions as a cofactor for catalysis. So this is the first phase, which is the carbon fixations, in which the Rubisco fix carbon dioxide into ribulose bisphosphate to form an unstable 6 carbon compound, which is immediately split into 2 molecules of phosphoglycerate, which is a 3 carbon compound. So this is the carbon fixations. Okay. So from here you can see the product of this reaction is an unstable 6 carbon intermediate with a 6 carbon compound, which immediately break down into two molecules of 3-phosphoglycerate, which is known as the PGA. Okay, PGA. Okay, acid A refers to acid. So with 3 carbons each. So the carbon of a carbon dioxide molecule is now part of the carbon skeleton in this organic acid. So we say that the carbon has been fixed. So that's why the names come from the carbon fixations. The Calvin cycle is also known as the C3 pathway because the first product of the carbon fixations is the 3-carbon compound, which is the phosphoglycerate. So that's why we call the Calvin cycle as a C3 pathway, because the first carbon fixations product is the 3-carbon compound. And then the plants that initially fix the carbons or carbon dioxide in this way are known as C3 plants. For the second phase, carbon reductions, they consist of 2 steps. That means there are 2 reactions here. The energy from ATP, which is produced from the light dependence reactions, is used to phosphorylate the PGA. Okay, phosphorylate the PGA into 1,3-bisphosphoglycerate. Okay, so 2 phosphate group here. So then the reducing power from the reduced NADP, okay, which is produced in the light-dependent reactions, is used to reduce the 1,3-bisphosphoglycerate into glyceroldehyde 3-phosphate, or we call G3P. Okay, so here's the first reactions, which is phosphorylation of the phosphoglycerate into the 1,3-bisphosphoglycerate, and then after that, the 1,3-bisphosphoglycerate is reduced into G3P. Okay. Then, as shown in the figure, in these diagrams, 1 carbon dioxide is fixed to produce 2 G3P. Okay, at the end 2 G3P. Okay, 1 carbon dioxide, so 2 G3P. Okay, so they're required to produce 1 glucose. After that, this 2 G3P, that means here they got 3 carbon, altogether is a 6 carbon. So the 6 carbon required to produce 1 glucose, which is also the 6 carbon. And at the same time, they are required to regenerate 1 RUBP, which is a 5 carbons. So can 1 carbon dioxide, which combines with a 5 carbons compound RUBP, so it becomes a 6 carbon. Can these 6 carbons can be used to form a 6 carbon glucose and a 5 carbon RUBP? So cannot. The answer is cannot. So therefore, the plants required 6 molecules of carbon dioxide. Okay, so carbon dioxide has 1 carbon, so they require 6 molecules of carbon dioxide. So must be fixed into 6 RUBP. Okay, 6 RUBP. 1 RUBP has 5 carbon, so with 6. So that means you have altogether is 30 carbons. So to produce 12 molecules of G3P. So 1 G3P has 3 carbons. So we have 12. That means here is altogether is 36 carbon. Okay, 36 carbon. So here is 1 plus 5 gets 6. 6 times 6 you get 36 carbon. And 36 carbon you can produce 12 molecule of G3P, which is also can give you 36 carbon as well. Then from these 12 molecules of G3P, 2 G3P molecules will be used to produce 1 glucose molecule. So from the 36 carbon, you minus 6 carbon, the remaining is, you still have 30 carbons. So the 30 carbons is divided, okay, divided by 10. Okay, so the 10 is remaining here. So then you divided and you rearrange the carbons, so what you can get is 6 molecules of RUBP. So altogether is 30. So that means in order to produce 1 molecule of glucose, the plant requires to fix 6 carbon dioxide into 6 molecules of RUBP. In other words, 6 Kelvin cycles are required. Okay, 6 Kelvin cycles are required to produce 1 glucose molecule. Okay, so the remaining here will be the 10 G3P. So through a series of 10 reactions that make up the regeneration of RUBP. That means stuff in the third phase of the regeneration of the RUBP. So here there are 10 reactions. However, the details we no need to know. Okay, and then these 30 carbons and their associated atoms are rearranged. Okay, so here you have 10 G3P. So the carbons, atoms, and the associated atoms. Okay, associated atoms mean hydrogen and oxygen. So are rearranged into 6 molecules of RUBP. And then the reactions is phosphorylated by ATP. Okay, by ATP. So these RUBP molecules begin the process of carbon dioxide fixations. Then the cycle is repeated. So as a result, 6 Kelvin cycles are required to produce 1 molecule of glucose. So therefore, 6 carbon dioxide have been fixed in order to produce 1 molecule of glucose. So this is a summary of photosynthesis. The light-dependent reaction, which is learned in the previous lesson, they occur in the thylakoid membrane, and the reactants that required and the products that form. Okay, so the hydrogen ions are used to form the reduced NADP, where the oxygen is released into the atmosphere. And the ATP and the reduced NADP that forms will be used for the light-independent reaction, which is known as Kelvin cycle. They occur in the stroma. And the reactants required is this one. And at the end of the Kelvin cycle, they produce the glucose, the RUBP, ADP, and oxidized NADP. So the reactions of 2 molecules of G3P leads to the formation of glucose and fructose. Okay, they can form glucose or form fructose. Sure, the glucose is used for cellular respiration. Then the glucose and the fructose are joined to produce sucrose in cytosol by condensation. Then the sucrose form will be translocated to other parts of plant. The glucose is also used to synthesize starch or cellulose. Then the starch form is stored in the starch grain of the chloroplast. The G3P can also be used to produce non-carbohydrate products such as amino acid, glycerol, and fatty acid. So for checklist, at the end of these lessons, you should be able to describe the light-independent reactions, Kelvin cycle, which has the 3 phases. The first one is carbon fixations. Second one is a carbon reduction. And the third one is the RUBP regenerations. So next lections, we will talk about the photorespirations. So what is photorespirations? See you on next lesson. Thank you. Bye.